Hello and welcome back to my second video for clinical neuromuscular intervention on synapse transmission. So if you remember from our first video, we talked about action potentials and how a stimulus, such as a sharp object, can poke somebody, and we have nociceptors or painful stimuli receptors that will send action potentials or signals, our way of communicating information within our body to our brain, to our spinal cord, to get a response to our muscles and whatnot. Is that these action potentials will travel to other neurons, and these action potentials are generated by a change from the resting membrane potential. So if we look at, if this was the first neuron that we were looking at in the first video, it is then going to synapse with this other neuron, and the first neuron is called the presynaptic neuron. The second neuron that is receiving the information from the presynaptic neuron is the postsynaptic neuron. And then right here I have this area blown up where you have the presynaptic neuron here and the postsynaptic neuron here, with the action potential traveling this direction or this direction here. So what happens is as this action potential arrives at the axon terminal here at the end of these presynaptic neuron, calcium is let in by voltage gated ion channels, and what this calcium is able to do is these small bubbles here, which represent little vesicles that have neurotransmitters in them, this calcium will then come into the presynaptic neuron and allow these and trigger these vesicles to go to the wall of the presynaptic neuron, and exocytosis, or basically just kind of a release or exit from the cell, and these neurotransmitters will come across what's called the synaptic cleft. As these neurotransmitters come across this synaptic cleft, they're going to diffuse across, and eventually some of them are going to make it to the postsynaptic neuron. Once this happens, these neurotransmitters, this area right here is blown up, so this is the synaptic cleft right here, and then this is the postsynaptic neuron. This neurotransmitter binds to ligand-gated ion channels, and this allows a change in the ions, so sodium or potassium, sodium to come in, potassium to eventually go out, and this can trigger a potential, a graded potential within this postsynaptic neuron. It can either be something that makes this postsynaptic neuron excitatory or inhibitory, and I'll talk about that here in just a second. And then as these, not all of these neurotransmitters will be used to go to the postsynaptic neuron. Some of them will just remain in the synaptic cleft, and some of them will actually go back into this area right here is the presynaptic neuron. This neurotransmitter will actually be reuptaked into the presynaptic neuron, or enzymes will come and degrade away the neurotransmitters, or they will just simply diffuse away from the synaptic cleft. So again, you have the transmission of a signal of these action potentials happening between two neurons because of the action potential arriving at the terminal, calcium coming in, this triggering the exocytosis of these vesicles to release neurotransmitters, the binding of these neurotransmitters to these ligand-gated channels for the release or for the influx of ions in and out of the neuron. And from here you get either, depending on the type of neurotransmitter that was released, you get an excitatory postsynaptic potential where the action potential is continued and the signal is sent, or an inhibitory postsynaptic potential where there's no signal sense, where it actually inhibits any information from being sent. And here is a little takeaway from what we just talked about, and I hope you enjoyed this video.